part is like. All right. Good afternoon, everyone. So my name is Frank Lynn, I'm the director of the Coke Ridge Center for Hearing and Public Health, and I'm really excited to welcome you to our first academic seminar series of the year. And we're really thrilled to have Jay Pinto visiting us from the University of Chicago. I'm going to go over some housekeeping issues first, and then I'll introduce Jay a little more formally. So just purely logistics, this is our hybrid seminar still. Most of a lot more in person, but we have a lot more online as well. If you are joining us in person, we'll hold questions at the very end. I'll moderate a Q&A at the very end. For those who are joining by Zoom, though, feel free to type any questions in the chat function, and Jennifer Deal here will help moderate those and ask us at the very end. We do have closed captioning. For those who are accessing online, you can click on the link there. They'll also post in the chat so you can access the captions. And then any technical difficulties, there'll be email address there. We'll also put that in the chat as well. And this seminar is being recorded. I'm really excited. This is the first seminar of the year, introduce our seminar speakers for this year, which for those of you who know what we do at the center, it's always a mix between, I would say, more classical sort of epidemiology and then getting into policy and then getting into sort of work in the industry field as well. So we're leading off today, obviously, with Jay Pinto. I'll introduce in a bit. Then next month, we have Kara Liu, which we're thrilled to have. She's a disability advocate, and she's the head of brand accessibility at Google, who works a lot around just overall accessibility issues around hearing, but other conditions as well. And she'll be joining us. That will be purely a virtual seminar for next month. She's not able to come in person. Then November, we're having Natasha Merton. Natasha Merton is an epidemiologist at University of Wisconsin. She's taken over the Beaver Dam set of studies from Karen Cruikshank. So classic trained epidemiologist, talking about some of the work she's doing there at the University of Wisconsin around multisensory function and cognition. Skipping December for obvious reasons. January then, we're thrilled to have Jeremy Walston and Peter Abadir. Many of you guys realize are geriatricians over at Bayview, and they'll be specifically talking about their new AITC collaborative, which is an aging incubator technology collaborative that's funded under a P50 grant at Hopkins. And they'll be talking about the work with the AITC and potential intersections with what we're doing at the center. February, we're thrilled to have David Green. So David Green, some of you may know, he won the MacArthur Fellow many years ago, social entrepreneur, has done a lot around basically sensory, namely vision, and as well as hearing, in terms of social entrepreneurship, starting new companies, and he'll be coming to talk. And finally, in March, Alejandro Lopez, who is an epidemiologist from Mexico, will be coming to talk to us about some of the work he's doing around occupational health and hearing. And finally, the last one this year will be our annual Cochlear Center Research Day. And we're thrilled to invite Devette Swempole. So Devette Swempole, some of you know their name, trained as trained as an audiologist, but also entrepreneur, has started a company called HearX, the technology which many of us actually use in our everyday research practices nowadays. And he'll be coming in person for that as well. So I should back up. All these seminars will all be in person this year, with the exception of Kara will be joining virtually, and Alejandro will be joining virtually as well. All right, so I'm thrilled to now introduce sort of Jay Pinto. So Jay, I've known for, I don't know, Jay, probably over a decade. And Jay did his undergraduate and medical degree at Stanford. Seems like probably many years ago now, probably at this point. But then later on did his residency and fellowship in otolaryngology, followed by radiology at the University of Chicago, where he's still now as a professor. At University of Chicago, like many academics, he wears many different hats, both as a clinician, overseeing the clinical program rhinology for the Department of Surgery, but then also wearing research hats in terms of being the Dean of Faculty Affairs, and then also being one of the lead investigators. Many of us know him through that role is through NSHAP, which is the National Social Health and Aging Project. Yeah, okay, NSHAP, sorry. A lot of Jay's work too focuses specifically on olfaction and how olfaction contributes potentially to other late life outcomes, in particular cognitive impairment, dementia, but then also looking more classic epidemiologically at risk factors for olfactory loss in adults as well. I think as I was reading your bio, Jay, I was really impressed with the acronym ARONOSE. So hopefully hear a little more about that. But Jay leads something called the ARONOSE Project, which is a mouthful, but it's actually really neat. Air pollution, Alzheimer's, dementia, neuropathologic, olfactory mechanisms, and multi-ethnic longitudinal cohort study, which is, I thought was a That was taking a while to come with the acronym, I'm guessing, but great. So I'm thrilled to invite Jay here to talk more about olfaction as a window to brain health. Thanks, Jay.
Great. Well, thanks for having me. And yes, I think all good studies have to have a good acronym. Hopefully, you're training everybody to think about good acronyms because studies with good acronyms get funded. So acronym is very important. So thanks for that very generous introduction. It's great to be here. I think it's really nice to be with a bunch of people who are really interested in sensory function and aging and outcomes, because that's what I think about a lot. And I'm going to share some of my work with you today. So I don't have to convince you that the classical senses are really important in life. They allow us to interact with other people and the world around us. And I think, just thinking about the pandemic, just being here in person and meeting with people, and I'm sure everybody being in person here at the School of Public Health, you really feel the difference in interacting with people, how much a difference that makes versus virtual or other environments. And so now I think the world is now aware of the fact that personal interaction is really important for us. And that personal interaction happens through these senses, so they're really critical. I think they're really key components of life and health, probably a little relatively understudied. Maybe we don't see that in our domain here, but if you compare it to sort of the vast world of research and health, I think they're a little bit understudied, and so we really need to understand how they are important for health, especially in older adults. So I'm going to focus on olfaction, and I hope that I encourage you to think about ways that not only the sense of smell is related to aspects of health and outcomes, but also how the other senses are related in the same ways or in different ways, and how those differences can tell us something about say the brain or aging or other aspects of our interactions. And there are things that are similar and there are things that are different. And then also, and this is not what I'm going to talk about today, but I know this is work that you're thinking about and working on, and others are how the senses are integrated and how they combine to affect those outcomes. Because I think multiple sensory impairments really have synergistic effects on health and personal interaction. So I'm going to start by just telling you a little bit about how the sense of smell works. I think that'll set the context for some of these other studies that I'm going to show you. And I think, first of all, I think olfaction is really critical to human quality of life. All of us in our day-to-day existence can think about how smells make a big difference to us. You can remember foods that your grandmother cooked. You can remember certain, the cologne of your father giving you a hug. Certain aspects of our life really have a tight connection to the quality of our lives. Perhaps, most importantly, it affects how we perceive flavor. So most of flavor is actually the sense of smell. So you can think about like a cup of coffee or the smell of cookies baking or the way that food tastes, and that affects you day after day after day. So really critical for sensing that pleasure of food and pleasure in other environments. It's intimately tied to memory, and I'll go over a little bit of the neuroanatomy, basic, neuroanatomy for me, dummies. And it's tied to centers of the brain that are important in memory. And that's why there's this thing called odor memory, where you remember that smell of cookies, or you remember your grandmother's house. You remember these things. It's really tied in there because it taps into those circuits in the brain. More on a basic level, it's really important for early warning systems in terms of detection of noxious substances. So the example I use frequently is, even a single-celled organism needs to know there's toxins over here, there's nutrients over there, right? And so you can imagine that those chemosensory systems are really evolutionarily ancient. And they're indeed an ancient part of our physiology ourselves. And I'll get into that in a little bit more detail, but really important for detecting toxins in the environment. So things like, in modern life, smoke, gas leaks, up to 30–40% of older adults cannot detect sulfur smell in warning odors in gas. So that's a huge safety issue right there, those two things. But even things like spoiled foods, things like detecting body odor, things like that, that really, are quite important for people. This is the way we detect it. And when it doesn't work, it's a big problem. Some of my colleagues at the University of Chicago have done some really amazing work in identifying the role of olfaction in kin recognition. So lower animals like mice can detect species differences and know which species to mate with based on odor of MHC proteins in urine. And there's always been a debate about whether that could be possible in humans. And my colleagues, Martha McClintock and Carol Ober in psychology and human genetics, looked at that question and actually found some evidence that that humans can actually do that too, that humans can detect odor among body odor of other people and can sense whether the people are more or less related to them and could have an influence in mating choice. Martha's also done some amazing work in pheromone detection and chemo signals that we give off that cause responses in other people. In a sort of tour de force as a college student showed that that chemo signals can synchronize menstrual cycles in a college dorm. This is work that she published in Nature in college. So really an inspiration to people who are, clever and thinking about their environment and keen observers of what's going on. And she's gone on to show that those chemo signals can have other psychological and physiological effects. It's important in mood and in emotion. Again, this is part of the neuroanatomy of the nervous system. I'll mention that we've done some work on looking at olfaction and mental health. And you can imagine that mental health would be involved in a lot of different sensory problems with aging. And also brain function in general, and we'll get to that too. One thing I thought was interesting I came across recently is that scented candles. So I don't know how many of you have candles or like candles, but that's a $4 billion business. So you can just imagine, like, people are so want to have pleasurable odors in their environment. The sense of smell is so important that people will spend $4 billion on scented candles. And so, obviously they're buying them for a reason, because it makes them feel good in their home or in their hotel or wherever. So that's just something to think about, like, how, where do these things make a difference? Well, you can see by the dollars spent on that alone. And that's not even factoring in the fact that big companies spend a lot of money thinking about flavor and odors and how it affects smell and products, and how that might be important in commerce and business in terms of getting people to buy things. And so, sorry, just to mention this potent wizard quote from Helen Keller. Really, you can think about how olfaction really transports you otherwise. You can close your eyes, you can, smell places, and it really brings back those emotions and memory. And that's a clear observation from the way that the brain is wired. All right, so how does this work? So, just in contrast to other senses, if you think about like how we understood about like, tonotopic, function of the cochlea and how we can perceive, sound, that's work that was done, say, in the 50s and 60s. And then if you think about vision, genes for color vision were discovered, say, in the 70s and 80s. And it's really, quite much later that we've discovered the mechanisms of olfaction. We're still working on some of these things. So Richard Axel and Linda Buck discovered this family of olfactory receptor genes in 1991. These genes are located on nearly all the chromosomes, I think 21 out of 23 chromosomes. So they're spread across the genome, indicating that they've been there a long time, and also indicating that they're involved in multiple different functions. And they discovered these genes, which are really a physical encoding basis for the sense of smell. So you have hundreds of olfactory receptor genes. Each one of them detects a certain shape of an odor in the olfactory neurons in the roof of your nose. Though the pattern of those different receptors, each of which responds to one shape of an odor molecule, will then signal a pattern of signals. That information is processed in the olfactory bulb and then transmitted onward to the brain. And then your brain interprets that as an odor and can tell very subtle differences. There's some very nice work that came out recently showing that, we think like, oh, our sense of smell is terrible, right? And if you think about like gene evolution, you think, well, olfactory genes, there's like about 800 or 1,000 genes, and only about 400 or 500 are functional. The rest are, we've lost them. They don't work anymore. They've stopped codons. So you think that sort of like, our olfaction is evolutionarily going away, and other things like vision and hearing are more recent and more important. But if you actually test people can detect a huge number of different odors. And if you do the math, when you do some nice experiments in humans where you give people differences and say, can you detect these differences? You can find that actually human sense of smell is very, very good and very good at discriminating different things, in particular mixtures. And the coding of we're still working on. It's very complicated. So all of which to say, we've got this, really amazing system for detecting odors, and it works through our nose. And these circuits, the information, unlike some of the other senses, goes straight into the brains. It's not the neurons don't synapse in the thalamus or other parts. They go straight into these different parts of the brain. And so what does that result in when it doesn't work? Well, it results in, things that are important to us. It impairs your ability to eat, to cook. It's hard to tell what flavors food has. You can't tell whether you need a bath or whether you've been to the gym. Do you need a shower now or later? It's hard to clean homes because you can't tell that there's odors. It's hard to buy fresh food. It's hard to socialize. So, you go out for meals and you enjoy the food together. It's a communal experience, and it's a social experience. And when you can't get pleasure out of that, someone says like, oh, doesn't this pasta taste delicious? And tastes like cardboard to you. Everything tastes the same. That kind of like has a big effect on how much you like to go out or be with other people. It's hard to detect spoiled foods, smoke, and gas leaks, as I mentioned. And then this results in symptoms of depression. So when you can't do these things, remember, sense of smell is directly tied to those pleasure centers, and depression by definition is an inability to sense pleasure. So you have this increased symptoms of depression that can affect nutrition, appetite, and psychological well-being, and overall just less strong quality of life. So, what causes these things? Well, the biggest thing is aging. And we know long ago from studies from Richard Doty, a nice paper in Science in 1984, plotting out odor identification and showing that it goes down with the sense of smell, with age, excuse me. And then the other common causes are trauma, so car accidents or surgery of that area, inflammation, and I include in there both things like chronic sinusitis and allergies, sort of immune system problems with the nose that are chronic. And then also in there is infectious causes like viruses, post-viral inflammation. And of course, we've seen that in COVID having a big effect. Other ones are neurodegenerative disorders, congenital problems, endocrine disorders, tumors of that area. So you can think about it, diseases in the nose, diseases of the nervous system, or systemic disease. You can also look at the airflow. These are odorants that are in air that are those odorants are wafting up to the roof of your nose where the olfactory receptors are. And so anything that blocks that airflow will cause problems. Also, if those nerve elements, the olfactory nerve is not working, so anything that makes the olfactory nerve not work will cause that. And then of course, problems on the central end with brain processing. And you can think about this for vision, you can think about this for hearing. It's the periphery and the center and the connection. And these are hard things to sort of tease out. We classify this as sort of anosmia, no sense of smell for all odors, or decreased sense of smell, hyposmia for general odors, or specific anosmias where people have a mutation in one particular olfactory receptor or a set of olfactory receptors where there's certain things they can't detect, there's no signal. So that's a specific anosmia that's fairly rare. There's also how the signal is processed and how you interpret it. So We have a huge number of people now who have what's called dysosmia or parosmia, where they smell a chocolate chip cookie and it smells like garbage, or they have a burning odor that's always present in their nose, even though it should smell different. And that's a huge problem for which we don't understand, but we think that it's related to abnormal processing of the signal. So when the nerve's damaged, it's sending some abnormal signals, and the brain interprets that as the wrong odor, and usually, interestingly, a distasteful odor. Phantosmia is a similar problem where the brain is sensing that something, they're giving you a signal that something's in the room, and there is no stimulus. And then finally, hyperosmia, which is fairly understudied, and maybe we should study it more, where you have increased sensitivity. And that can be just as much of a problem as decreased sense of smell, because there's some things you don't want to smell all the time. If you can smell something in the corner, there's a dirty diaper in the garbage 100 yards away, that is irritating and can be quite much of a problem. So what are the big causes? I mentioned age, and most of the work I do, all of the work I do is on aging. But there are other upper respiratory infections like SARS coronavirus 2, inflammatory diseases like rhinosinusitis, trauma, which is usually pretty obvious, surgery, which is pretty obvious, Alzheimer's disease, Parkinson's disease, and other neurodegenerative disorders. Little more rare is chemical injury, toxin exposure, endocrine diseases, and tumors. And usually those are relatively obvious in terms of a clinical point of view. So how do you measure this? And this is another thing to think about, like when you're measuring a sense, like there's lots of different ways to measure it, and what's the right way to capture the physiology or the outcomes that you're interested in. In this case, most people are unaware of their sensory deficit of olfaction, and so objective testing is important. I think it is worth considering, though, that even survey data can capture information, even if it's not exactly what you're interested in. So it's always worth thinking about, like, what are you interested in? If you're interested in how something impacts people, then you are interested in the perception, whether that's related to the objective measures or not. It's also worth thinking about why do people perceive certain sensory deficits in a way that doesn't correlate with the testing? And I think that's another interesting area to think about. But testing allows assessment of the degree of loss. It allows for monitoring change over time. And then clinically, we use this to establish validity of disease. So I have patients who come and see me, they say, I have a poor sense of smell. And really what they have is airflow problems. So they have blockage of their nose. And if I can fix that, they perceive that their sense of smell is better. Even though if I test them, they can do just fine. Rarely we have patients who come in saying, I had XYZ injury or exposure, and now I have poor sense of smell. We want to validate that for insurance reasons or other reasons. And so that is where testing comes in handy. We can measure this by odor identification. So here's an odor. What does it smell like? You get some choices, and that's commonly done by the University of Pennsylvania Smell Identification Test. You can do it by marker-based methods, where there's a marker with an odor, present it, and again, have a word or picture choices and get an answer. And we use that. That's nice because it's reusable. And then there are cross-cultural versions. The interesting thing is, no matter how you test odor identification, and if you test it in Sweden or Africa or different countries and different tests, you see similar relationships, which tells you that modality of testing, however you do it, is capturing that same information. And there are cross-cultural versions of all these tests, and they perform pretty well regardless. We can also do threshold testing. So that's how sensitive are you? So if I give you something at a low concentration, can you detect it? And then discrimination. Here's two odors. Can you tell if I'm giving you two of the same odor or two different odors? Can you discriminate those two odors? And finally, you can do neurophysiologic techniques. Put electrodes on the head, present an odor. Do you see a signal? You can also do that by fMRI and other methods. So a lot of different ways, but again, you have to think about what's practical. So some of these things are not practical for clinical practice. Many of these things are not really practical even for big population studies. And so we have to rely on things that we can get to work on a practical basis. All right, so with that sort of background, there is a lot of work done on the sense of smell in large population studies. I mentioned that original work in a cohort of patients at the University of Pennsylvania. And since then, there's been a lot of interest in that. So for example, there was a large study in the late 80s, early 90s, National Geographic Society. So National Geographic had this magazine, for those of us in the audience old enough to remember, and they sent out this magazine, and like millions of people across the world got this magazine, and they decided to say, hey, Richard Doty published the scratch and sniff smell test, let's add one and put it in our magazine, and we'll send it out all over the world, and people will, I guess, mail it back, and we'll find out what people's sense of smell are. So they did this, and it was a great study. I mean, it was a really amazing study. I think they got 2 million people to send it back. So this huge study of sense of smell, got some very interesting findings from that. And now sense of smell has been incorporated in, and I won't read all the studies here. I bet many of you work on some of these studies. You know, so you have a number of different data sets to look at the sense of smell, and depending on the topic of the study, then you learn different things about olfaction related to, say, you know, atherosclerosis or ethnic variation or relationship with other senses, et cetera. And so these are some of the studies that are out there, and there are probably others. But it's something to think about when you're interested in sensory health and aging is, you know, who's collecting this data, how are they collecting the data, and what other health information you can get out of it. So the study that I'm part of, lucky to be part of, is the National Social Life Health and Aging Project. This is a study led by Linda Waite at the University of Chicago. She's a sociologist and aging researcher, and we have a very multidisciplinary team to work on that. So we have psychologists and health economists and geriatricians and diabetologists and people who work on social health and some very nice collaborative group. It's a study that started in 2005 with a probability sample of 3,005 community residing adults, meaning they live at home, ages 57 to 85 at baseline. It's a sampling frame that we borrowed from the HRS. It's representative of older adults in that situation in the US population. And we've used it to study a whole number of interesting things related to things like elder abuse and social disconnectedness, sexuality, smoking and alcohol use, medication use, diabetes, frailty, sleep. There's just a huge number of topics that have been, this data set's been used to address. And to give you an example, there was one colleague we have, Dee Mikado, who's a pharmacologist, did a very simple study of looking at, okay, here are the medicines that older people tell us they're taking. How many people are on medicines that are in conflict, that they're not supposed to be on together? And she found that a huge percentage of people are on medicines that they're not supposed to take together. So, you know, very practical and very impactful work. Stacy Lindau, who's an obstetrician who's part of the project, did some very nice work published in the New England Journal saying, like, you know, what is the landscape of sexual function of older adults? And she found some very interesting and very relevant, you know, findings about what people care about, what people are doing, what their practices are, you know, really informing how we think about sexuality in older people. The nice thing about this project is we have detailed and standardized health questionnaire data, like many studies. And one thing that's a little unusual is we have measured all 5 senses at the baseline. So we have measures of vision and hearing and touch and taste and smell. And I'm going to talk about smell today, but you should keep in the back of your head that we have some other measures that you might be interested in. And then we have multiple waves of data collection. So we followed the data, original subjects and their partners 5 years later. Then in 2015, we followed all those people and added a new cohort of baby boomers who were born later. And now we're in the field right now collecting the 15-year follow-up. So we have longitudinal data on these subjects, and that's another advantage. So how do we measure the sense of smell? So we use those markers, and we developed, based on some prior work, a 5-item shortened battery of what they're called sniffing sticks. Again, we give these people a marker. We say, what do you think this smells like? We have a picture and some words. They pick what they think it smells like. It takes 3 minutes to complete. So again, in field studies, you got to think about what's the survey burden, what's the respondent burden. So short burden. And then we classify these people into either anosmia or hyposmia or normosmia based on the number of items they get correct. So we can get a pretty good sense. We can categorize people, and then we can see what their sense of smell is like. I'm going to show you talk to you a little bit about cognition. So we have a cognitive assessment in NCHAP at the baseline. In 2005, we measured it with the SORT Portable Mental Status Questionnaire, which is really a screening tool. And then after that, we switched to the modified Montreal cognitive assessment, a modified version of the Montreal cognitive assessment, or the MoCA. And that's a little better tool, a little more discriminatory, has different domains. Both of these are appropriate for a national in-home survey where we can't do neuropsychological testing. We can't do heavy duty battery of testing. This is a sort of a 2-hour survey. So, we can't have, 45 minutes or an hour of that be the cognitive function. Again, we have this multidisciplinary group. And so everybody wants to get their part of the survey in. And then we can divide that into subdomains and look at different parts of the brain and its function and sort of figure out if things are related to one part or another, one function or another. So we have other information, including obviously demographic factors, physical health, comorbidity, mental health, smoking and alcohol use, and then education. We also have assets, which is useful rather than income, because these are older people. And so how much wealth they have is maybe more important than how much income. Their income might be low, but they may live, and retired in a house that they own, for example. And, we can ask the question, how does olfactory function here in this cartoon, coming in through the nose, synapsing in the olfactory bulb, going on through the olfactory tract, and then getting the things like the entorhinal cortex and the hippocampus and the amygdala and the orbital frontal cortex, these different parts of the brain that have different functions, really important ancient functions like memory and emotion and well-being, and how does it affect those functions through this system. So many studies have shown that olfaction is connected to cognition, and we wanted to ask the question, could this be useful as a clinical tool? And in fact, there are trials right now going on to see if you have odor identification, does that help people categorize people who are in high risk, medium risk, low risk for Alzheimer's disease? So could clinicians use this as a tool? And we were able to show that people with olfactory dysfunction had 2 times the odds of developing dementia 5 years later, adjusting for baseline cognitive function, age, gender, race, education, and comorbidity. So a pretty good predictor of who's going to have problems with their brain 5 years later. And again, partially because the olfactory system is an ancient part of the brain. I think about it as like a part of the brain that's in your nose. It's right out there in the world. And I'm not going to talk about pollution today. I wish I had included some of that. So pollution is one impact that I'm studying, but also it's a canary in the coal mine for how well the brain's working. So when the sense of smell starts to not work, it's an early indicator that the brain is not going to work later. And in fact, you can find Alzheimer's pathology in the olfactory bulb in patients before they get Alzheimer's pathology in other parts of the brain. And we don't really know why that is. And we're going to, people are working on that question actively. So the next question is, could olfactory function predict other aspects of brain function? So because we're very interested in sort of change in olfaction, you can imagine that if we measure any parameter at one period of time, there could be all kinds of things affecting it. But how about how that sensory parameter changes over time could give you some difference, and the trajectory of your sense of smell could be important as a predictor. Maybe people who are falling off quickly are different than people who are slowly declining. And of course, we're very interested in the people who stay constant over time. So could olfactory trajectory predict brain health outcomes? So for this question, we worked with colleagues. David Bennett runs the Rush Alzheimer's Disease Center in Chicago, and was really a pioneer in Alzheimer's research. And that in 1991, he started a cohort of people and followed them over time with annual visits. Detailed annual visits where you can do very detailed neuropsychologic testing, and then slowly has added on over time many different other measures that he's used to do some amazing work about how does dementia develop. And in fact, another interesting thing is that he collects the brain and spinal cord from these patients when they die, so then has done some very nice work on the pathology and how do clinical predictors beforehand end up with pathology in the brain related to Alzheimer's. So it's a longitudinal study, annual evaluations. People didn't have dementia at baseline, and then follows people, does clinical diagnosis of dementia, and then you can look at how things change over time. And one of the things he collected was a sense of smell. I should mention, for those who are interested, he has actigraphy and structural and resting state fMRI, well-being, behavioral economics, gene variation, epigenetics, many different aspects. So for those who are interested in those parameters, he's doing a lot of amazing work, and he's extremely generous to collaborate with. So his olfactory test, again, was the 12-item brief smell identification test. It's a version of the UPSIT, and it's a 4 alternative forced choice tests, just like our test and just like the main UPSIT. And then the cognitive battery was 19 cognitive tests. They average the scores and make a Z-score out of that for what's called a global cognition score. They can subdomain that, divide that in 5 domains. And then there's clinical adjudication, where you can divide people into those who have normal cognition, those who have mild cognitive impairment, and those who have Alzheimer's disease. So the imaging that they have is 3T MRI. It's segmented and processed using standard methods. And then you can get gray matter volume. So, how big is a region of the brain as a percentage of the overall volume of the brain, so sort of normalized to brain size. And then you can divide those regions up into different parts of the brain, like the primary olfactory region, the secondary olfactory region, and then other regions such as temporal, frontal, parietal, visual, or sensory motor. And so I had a amazing medical student who had a lot of expertise in neuroimaging, and she really wanted to take this project on and ask the question: if we look at change in olfaction over time, does that predict who's going to end up with those cognitive categories, and does it predict structural changes in the brain? So this is our study design. Again, I mentioned we have this annual olfactory testing, and we have these time points where people are tested, and then they get MRI, and then eventually they have a final clinical diagnosis of either Alzheimer's and mild cognitive impairment or dementia. And I should mention that we decided to structure the study where the repeat olfactory testing, we chose the period where people have normal cognition. So these are people who have normal cognition, but their sense of smell is being tested. We can divide people into, are they changing, are they, how they're changing over time, and then look at the outcomes, both in the structure of the brain and then their clinical diagnosis. So this is an older cohort. The mean age is about 76. We had MRI only on 121 of the people due to that sort of selection criteria. Mostly female, mostly white. Pretty well educated, and the baseline odor identification score was 10 out of median of 10 out of 12. So pretty good olfaction to start. We have quite a number of years of follow-up, so somewhere between 7 and 6 years of follow-up, so we can sort of plot their trajectories. I should mention that, and I don't have this slide, but when we plotted this, it's all over the map. Like, you just have a spaghetti map of people, and we ended up having to sort of reduce the information burden there by creating slopes of those lines. And then we also have ApoE genotype. And as you may know, ApoE4 is a risk factor allele for Alzheimer's. So we can account for genetic risk here. So we were able to show that a faster olfactory decline did predict overall worse global cognition. Sorry, this slide is somehow not animating. I guess I'll just tell you that, and there were particular functions of the brain that were affected and others that were not affected. So as you might imagine, as I alluded to earlier, episodic memory was really close. Memory, some aspects of memory, like episodic memory, was very closely tied to olfactory decline. Same for perceptual speed, and less so for semantic memory, working memory, and visitor spatial orientation. So we can see different olfaction. This sense is related to certain parts of brain function, and maybe other senses might be related to other parts of brain function. And interestingly, we were able to show that olfactory decline also has 2 times the odds of being diagnosed with mild cognitive impairment or dementia. So this is on the order of ApoE4 alleles, like one of the big risk factors for these cognitive outcomes, indicating that this simple clinical test, just following people's sense of smell over time, can give just as much information. Now, when we looked at the MRI, we found that olfactory decline predicted gray matter volume loss in olfactory regions. And perhaps this is not surprising. Your peripheral sense of smell, say, is declining. And so the brain, the neurons in the brain that process that information are shrinking. And we see a nice trend here with the more severe decline, the smaller your brain volumes are, primary olfactory cortex, also the secondary olfactory cortex. And what was interesting was that not in other parts of the brain. So this wasn't just all parts of the brain are aging and decline. This isn't an indicator of just sort of overall brain shrinkage. This is specific to certain parts of the brain and not other parts of the brain. So not in the visual cortex, not in the sensory motor cortex, was very specific. The other thing that we found was interesting was that olfactory decline predicted gray matter loss could distinguish that you could see the differences between people with normal cognition, mild cognitive impairment, and Alzheimer's disease. So you can see here that patients with Alzheimer's disease have a smaller primary olfactory region compared to the other categories, also in the secondary regions, and again, not in the control regions. And so these patterns that we see in the olfactory areas seem to be specific to those with neurodegenerative disease. So olfactory decline is a good predictor of key features of Alzheimer's disease. It can predict interval diagnosis of dementia 5 years later. Maybe that could be useful clinically. People who are cognitively normal and have faster rates of olfactory decline are more likely to develop impaired cognition and dementia. They suffer from structural gray matter, smaller areas in their olfactory brain regions, and these patterns are similar patterns that we see in mild cognitive impairment and dementia. And so serial assessment of olfactory function to monitor rate of decline could be a tool to identify people at high risk. And you might say, and I get this sometimes in reviews, they say, well, who cares? Like, we don't have any treatments for this. But I think that's a sort of a short-sighted thing. First of all, we can identify people for clinical trials. We can do earlier interventions, whatever kind of intervention that is, pharmacological, diet, exercise, many different things we can start earlier. Number two, we can inform those families, right? So if you knew that your loved one was going to have problems 5-10 years later, you could start to plan. And obviously caregiving is a huge burden for these families. What about our other, so I'll just finish now by talking about other aspects of physiology. So the title of the talk was, cognition, and I'm not the first one to look at olfaction and cognition by any means, and I think it's a really exciting area to work in. But what about other aspects of human physiology? So, I mentioned that olfactory receptors are spread across the genome, and indeed they're actually expressed in all kinds of other parts of the body, and we really don't know what they're doing there. So, for example, olfactory receptors are expressed in the lung, they're expressed in the gut, they're expressed in the kidney, they're expressed in the testes. And we know that these are G proteins that can signal based on chemo signals in the environment. So they're picking up molecules and they're sending a signal. What are they signaling? What are they picking up? And what is that doing? It's at least in the upper airway, I can sort of understand, right? So these are chemosensors in the nose. If you smell like this room's on fire, your nose tells you that, your brain tells you that. It's an airway function, right? It's sampling air as it goes through the nose into the lungs. So it's pretty easy to tell people, hey, this could be involved in breathing diseases, particularly the upper airway, but also the lower airway. But how about these other functions? Could it be involved in nutrition and renal function and sexuality? So for this, we turn to the Health ABC study, Health Aging Body Composition Study. You probably know this data set better than me, but this is a study of where they did annual clinic or home visits, telephone follow-up after that, and then measured sense of smell with the 12-item upset, just like in the Memory and Aging Project, and categorized that in a very standard way. And this is a work that I did that really Honglei Chen led at Michigan State. He's a collaborator, and looked at the outcome. Okay, could olfaction predict other airway disease outcomes. And in this case, they looked at hospitalization due to pneumonia, both total hospitalizations and then first ever hospitalizations related to pneumonia. And so this is about 2,500 participants, again, older people, about half men and women, about 40% African Americans. They had good numbers of people with both poor, moderate, or good olfaction. It's like pretty amazing that the data set turned out to be about a third in each group. And about 30% of people had a history of pneumonia in the past, about 25% had a history of kidney disease in the past, about 25% had a history of hospitalization in the past. So you have enough discrimination to sort of detect some associations. They have about 12 years of follow-up, and there were 801 hospitalizations due to pneumonia from 500 of these participants. Most of that was for the first time. So the interesting thing is we found that older adults with poor olfaction had increased incident pneumonia hospitalizations. You see here in this chart that those with poor olfactory function had a higher cumulative intensity of having a hospitalization. And you can see it very nicely here in the rate per 1,000 person years of being admitted. And you can see here that people with the poor olfaction group, right here, have a much higher rate of hospitalization. And indeed that those with poor versus good olfaction had a much higher rate of total pneumonia hospitalizations. Poor olfaction was also associated with a higher rate of first ever pneumonia hospitalization. These were similar across sex or race groups and adjusted for a number of factors that could be relevant for hospitalization. Of course, obviously hospitalization is a tricky thing as an outcome because many things can affect hospitalization. But as you can see here, we've accounted for things like prior hospitalizations, history of pneumonia, history of lung diseases, and a number of other different things. So why would this be? This is like such an interesting finding. And I think it could be that it's an indicator of loss of barrier integrity. So when your upper airway, when your airway is connected from your nose to your lungs, and when you have loss of the barrier, pathogens can get in and cause pneumonia. I also think it could be related to the sensor function. So those olfactory receptors are in the nose. And if you come to the room and you can't detect smoke and you breathe smoke a lot, that's eventually going to hurt your lungs and predispose you to disease. This could be true for pathogens. And there are chemosensors in the nose that detect molecules that are produced by pathogens or airway toxins. It also could be an airway sensitivity. So when your nervous system can't detect chemicals or things in the environment, you cannot sense what's going on, and you might have something called aspiration pneumonia, where things go into your lungs from swallowing that aren't supposed to be there. So it could be deterioration of chemosensoration in the lower airway, and that's something we need to work on to figure out these mechanisms. It's pretty interesting that something that's totally you wouldn't think is related to lung function or lung outcomes predicts lung outcomes. Okay, what about chronic kidney disease? Again, this is the same data set, and depending on the analysis, we have between 1,500 and 2,500 people. They estimated kidney function, a parameter called the glomerular filtration rate, standard measure of kidney function, and then they defined kidney disease by standard measures by categorizing that and decline of that. And then looked at hospitalization and death in a similar way. We did methods to look for other causes of death, other reasons that might affect the risk of death. And it turned out that people with anosmia had worse kidney function than those with good olfaction, and people with decreased olfaction had worse kidney function. Those with anosmia at baseline had worse kidney function 7 years later, although when we adjusted for some other factors, that became a trend. And it wasn't associated with physician diagnosis of kidney disease or hospitalization. So olfaction or chemosensation seems to be involved in predicting who's going to get pneumonia, but it doesn't predict who's going to get kidney disease. And again, these were adjusted for a number of relevant factors. So what can the mechanisms here be? Well, it could be that even early on, uremia, the toxic byproducts of not having your kidneys work, could affect the sense of smell. And these toxins may affect nerve function and neuroprogenitors in the sense of smell. These things are expressed in the kidney, so there could be something going on in terms of the kidney itself. And interestingly, some hospital-based studies show that people with poor kidney function have worse odor identification. And it's also associated, kidney disease is also associated with 3.8 times the odds of having poor odor identification in some adjusted analysis. So seems to be a relationship here, a little harder to explain. How about nutrition? So you can imagine if you can't smell, you can't taste flavor, you're gonna have malnutrition. It's actually also been associated with obesity, maybe because if you can't taste flavors, you can't get sated, and so you eat more. You're like you want to be satisfied, you can't be satisfied. And we have shown that poor olfaction leads to weight loss in older adults in NSHAP. And so in this study, we looked at the question of could it be involved in body mass? Again, turning to the Health ABC, where we have body mass measured by some standard measures, we can look at this in cross-section and over time to see how sense of smell predicts body mass. And it turns out that poor olfaction is associated with decreased weight and weight loss. Those with poor olfaction have way less, and that's both true for total body mass, lean body mass, and fat body mass. In longitudinal analysis, those with poor olfaction had greater annual decline for both total body mass and lean body mass, and even a trend towards decreased fat mass. These are what these graphs look like. So you can see you have the normal and decreased olfaction here, and then anosmia here, and you can see some pretty clear trends for total body mass, lean body mass, and maybe not quite as clear for fat, but maybe a trend there too. So how could this be? Well, this is a little easier to explain. May alter your appetite, dietary intake. It's linked to hormones like leptin, which is involved in satiety and food intake and metabolic rate. And mice who have a poor sense of smell do lose weight, even when you hold calories constant. So it's not just eating less, but somehow it's related to metabolism, which is really interesting. And it has big implications because we know that just weight makes a big difference. And in fact, we've done some work at NSHAP showing that obviously there's a little bit of a curve, but like fatter people actually, after you live a long time, actually end up being healthier, maybe related to bone mass or other things. And so keeping your weight up is actually quite important for older adults. Finally, sexuality. We measured in NSHAP sensitivity to butanol and odor identification. We have some really detailed questions about sexual activity, both frequency of thoughts and activity and satisfaction. And we're able to show that decreased olfactory function was associated with decreased sexual motivation and decreased emotional satisfaction, but no relationship with decreased frequency of sex or physical pleasure, interestingly. And that contrasts some other work we've done where we looked at global sensory impairment across all 5 senses, showing that global sensory impairment is associated with decreased frequency of sex, but not decreased frequency of thinking about sex or satisfaction with sex. We have this sort of paradox where people with sense of smell problems, they have sex with the same frequency, but they think about it less and they're not satisfied. Whereas people with global sensory impairment, it's more important for initiating and performing the sexual acts, but not really thinking about it or being satisfied by it. Again, maybe this is related to that connection I alluded to at the beginning, which is limbic system, emotion and memory and pleasure, like really tied in there for sense of smell. So it's critical for sexual attraction for men and women, including in older adults. And this is another area that I think is really interesting, like what's going on here, a really interesting part of physiology. So I'll just end for just a moment by mentioning COVID. So, this is in the New York Times, you see this all over the place. We know that SARS coronavirus 2 affects the sense of smell. I'm interested in actually the other question is, what can COVID teach about the sense of smell? But also now we have a virus which affects the sense of smell and hearing and maybe balance too, incidentally. And now we have a sort of an acquired cause, and we can see what happens over time to these people, right? So I've just been talking about work that's aging. As best we can tell, this is what we're studying age-related smell loss. But now we have acquired smell loss. So the question is, does acquired smell loss result in some of the same kind of problems that I just showed you was going on in aging? And we have a natural experiment to sort of try to figure that out. And so that's like an open opportunity. This is thousands of people, millions of people all over the world are potentially affected by this. And so this is something we're going to have to study in the future. And, I don't know how you all feel about long COVID and what it is and how it really affects people, but these are questions we're going to have to answer as a field, and particularly in the senses. we will be able to do that. So, I'm interested in sort of the opposite is how will COVID change the role? How will COVID change sensory function and aging over time? And now is the time to start thinking about that. We have a lot of bad data about COVID all over the place. Now we need some good data about COVID, sequelae of COVID, especially in sensory function and aging, and you're really kind of poised here to do that. So that's something to think about in the future as you plan your careers. So thinking about abnormal changes in sensory function over time in older adults can yield clues about human health and disease across a number of different domains, not just neurologic outcomes like ADRD, but also other aspects of human physiology that are really like underexplored. And these relationships are really important clinically, and maybe we could use them to mitigate the public health impact of these prevalent geriatric conditions. So things like kidney loss and pneumonia and nutrition, weight loss. These are big problems for older adults. And so if we can use some of these things that we've learned about them to prevent those issues, flag these people, that can be very helpful. And we're really at the infancy of studying these areas, and we could use that information to sort of help patients in a major way if we can get there. So, I've told you about the sense of smell, but think about how this could be important for hearing or taste or vision or touch. Really want to start looking at the senses together. How are they different? How are they similar? And how do those differences and similarities affect health and aging of older adults? So with that, I'll conclude. I'd just like to acknowledge the people who did the work here. So Rachel Pacina did the work on brain structure and olfaction. Jesse Siegel did the work on sexuality. I have a wonderful graduate student, Selena Zong, who also did some work on global sensory function and sexuality. Dara Adams is one of our graduates of our residency, and she's a rhinology fellow at Harvard now, and she did the work on olfaction and dementia. And then Honglei Chen led the work on those other health outcomes, including his wonderful students, Yaquan, Frank, and Karen, who led those relevant studies there. So thank you very much. I really appreciate the invitation to speak here. It's been a wonderful day so far. I'll mention that NSHEP is a publicly available data source. You can get it on the NACDA at the University of Michigan. I'm happy to help anybody navigate that process, or if you have questions about the data, it's a great data set. We want as many people to use it as possible. Thank you very much.
Jay, thank you so much for the talk, which I love the idea of spanning your overview of the biology all the way to functional applications, which is you don't always hear in all talks, and it's wonderful to see that. I want to open up to the audience. We've got 3 or 4 minutes for questions. Any questions from the audience? Dr. Sculo?
That was a sensational presentation.
Thank you.
Wonderful presentation. Congratulations. I have a couple of questions. One is, factory decline or loss may happen suddenly. For example, after neurosurgeon, brain surgeon. Or it may happen more slowly, like age-related and so on. Do those both have a relationship with cognition?
Yeah, it's a great question. So it's really not been studied very well. So people have tried to study quality of life after acute smell loss, say car accidents, and you can certainly find relationships with things like depression and nutrition and some other parameters that are pretty obvious. What hasn't been asked and hasn't been studied as much is what's the effect on the brain. And so I'm very interested in this idea that cognitive load, like your brain is getting signals, in this case olfactory signals, and it's keeping those neurons alive, right? They're functioning, they're growing, they're making connections. And when you cut off that signal acutely, for example, how does your brain react? You've got to imagine that decreased input is going to have an effect on the brain. And it's really, really understudied. We don't know the answer to that. What I also will say is the brain is quite plastic. So we know, for example, from stroke that if one part of the brain doesn't work, you can actually train other parts of the brain to work. And we don't know if there's other parts of the brain that compensate for that lack of sensory input that could actually affect that. So it's a great question. I'm very interested in that question. There's been some early data about COVID where it's sort of acute, right? You have an infection that kind of kills your sense of smell. And there is some evidence that there's some cognitive problems in MRI and function after that. Now, that data is very difficult because many of those patients have had other problems. So they've been in the ICU, they've been on pressors, they've had pneumonias, they've had other healthcare related problems that kind of confound that relationship. But we now have people who have, many people, myself included, who have had COVID, did not get admitted to the ICU, did not get any other treatments. And now we can sort of see, like, in that, mild category, does that have any after effects? And we can see, if we see the same problems with acquired or acute smell loss that we see in age-related smell loss. And I don't know, Frank, if you know anything like related to hearing, like sudden sensory neural hearing loss or other things, you might be able to comment about that.
It's, I mean, for COVID and hearing, it's questionable at best, right? I mean, it's on a background of sudden hearing loss, sudden something about 1 in 10,000, and we see that being roughly about the same. So it's jury's still out on that one, but it's not an extreme effect though, unlike olfaction, which is very obvious very early on. I want to be mindful of time is 1:01. So I want to thank everyone online for joining us, everyone here for joining us. And Jay, thank you for really kicking off our academic seminar series here in person. It's been it's nice getting back to normal per se. So thank you. I think for a lot of the trainees, you guys will have lunch with Jay now too. So I think there'll be a lot more questions, but it'll be during the lunch session. We'll
I'm sorry I went long, but I would be happy to answer questions afterwards in the discussion next.
Thanks again, Jay.